some people say the golden
trade desires, and that piece of the game.
Guys, you guys have to be quiet now. Van?
Yeah, just one battery.
What kind of batteries do you need?
Double A.
What do you want? I want change.
What do you need?
Please.
What do you got?
That's only enough that I can find.
That you feel the same. Despite all my rage, I'm still just a rat in a cage. Someone will say what is lost can never be saved. Despite all my rage, I'm still just a rat in a your scaffold if I
Welcome to MetPhys 2020, Metabolic Physiology in Isolation. This is a new model for truly inclusive global science. These meetings online are cost-effective, inclusive. Anybody can listen to them anytime, anywhere. And there is no cost, in terms of carbon emissions from flights, people having to travel across the world to get to these meetings. Yet, we're still managing to get you some of the best stories in metabolic physiology today. We've been really enjoying the action online on Twitter using the MetPhys 2020 hashtag. Cassidy, now is your time. You have managed to take a photo of yourself in front of the screen at this time every day so far. This is day 10, and yet we've had 9 photos in your Inception Challenge. Keep it going. Can't wait to see that photo. I bet it's already online right now. Thank you, Rob, for taking those photos every day. There has become a new category in the MetPhys 2020 challenges. I thought Felix Vogel was going to take the lead on this. He looked like the strong front runner, riding his cycle while also listening to MetPhys 2020 talks, until on the weekend, tomorrow's speaker launched his own attack on this title with a new treadmill Pac-Man game where he feeds his kids marshmallows on a treadmill to keep them occupied. I think that's a fantastic idea. I think that's also in the lead for the best way to keep your kids occupied. If you've got a treadmill at home and some marshmallows, why not try it? It'll keep them quiet at least for a few seconds. Trick, slow down the treadmill. All right, moving forward, I've said this every day, I really mean it. Please take note of all these faces. These are the wonderful, incredible colleagues of ours that stood forward very quickly and said, I will give a talk, I will share my unpublished work to keep everyone entertained, to do our part to help flatten the curve and keep everyone home, at least for an hour a day. So remember these faces, get on Twitter, ask some questions after their talks, email them. If you see them in person, thank them. If you're at a coffee house, buy them a coffee. If you're at the bar, buy them a drink. Let them know how much you enjoyed their talk. I know that'll mean a lot to them. I've got wonderful news. You're going to have to remember some more faces. We are extending out to April 24. That's going to be the end of it. After that, I'm going to have to take a break. But there are some fantastic additional speakers that have agreed to fill out the schedule up to April 24th. That's it. Once daily talks at the same time with the same Zoom links. I'm very excited about listening to these talks. I'm sure you will be too. So stay tuned, please. If you'd like to thank me personally, I wouldn't be here without the Pathway to Stop Diabetes program from the American Diabetes Association. That's really got my lab off the ground and running, allowed me to do the science that I really am passionate about doing. If you have $5 to spare and you want to make a donation, that would really honor me, but you don't need to do this to be part of these talks or to listen. It's available to all. For you people who have friends who are not on Twitter, who are unaware of this, feel free to share the links. If you share the links to the White Megara Lab webpage, we have links not only to the Zoom, but also to the Facebook page where the talks are streaming live and then become recordings, and also YouTube, where you can search in the search space hashtag MetPhys 2020, and usually by a couple of hours after a talk is given, those talks will be up there. If you can't find them on YouTube and you can't find them on Facebook, please email me. I'll figure out a solution. But in the meantime, please enjoy the talks. Today's talk from Gareth is going to be exceptional, and tomorrow is going to be great too. Tomorrow we have my colleague, John Campbell from the Duke Molecular Physiology Institute, giving what should be a fantastic talk about expanding the definition of the incretin axis. But right now I'm going to pass over to Gareth. Gareth, I'm getting ready to pass you the host controls. So please enjoy, and don't forget to stay around for the Twitter question session. Please don't click on the raise hand button during the talk. The speaker is only going to answer questions in the Twitter session afterwards, or if you email them personally. once. Let's make Gareth the host. Yes.
Okay. Well, thanks, Phil, for the introduction and for organizing this. Hello, everyone. It's a great pleasure today to be able to present you some of the work that my lab has been up to for really the last 3.5 years. Here we go. So once again, thanks to Phil, and also thanks to all the speakers and the attendees for your attention, and also for participating in these in this great series of talks. I'm learning a lot, and I'm sure everyone else is as well. So I like to start my talk really just with this simple cartoon diagram. And it's a, you know, whenever we open up a textbook or review paper and we look at signaling pathways, often it's reduced down to very, very simplistic, simplistic format. You know, so for example, if we take this rectangle here or as a receptor, you know, binding of the ligand to the receptor elicits a response, as shown here by this gray circle. But, you know, this is probably the most simple way you can think of a signaling pathway. You can start to throw in other effectors, you know, downstream or in between the receptor and the outcome. And because cells are not two-dimensional, you also have to consider the concept of space or the cellular compartment. And, you know, this further complicates things, right? And multiple parallel pathways can also be activated to elicit the same outcome, as well as modulators that can play a part. So really, cell signaling is not as simple as we like it to be. I'm sure all of us can appreciate that. And you know, the question that has really been driving my lab's research is what factors ensure the accurate coordination of these signaling effectors and their activities. Just to drive this point home a bit, this is a cartoon diagram of the insulin signaling pathway. And as you can see, it is very complicated, whereby activation of the insulin receptor can regulate glucose uptake, glucose synthesis, gluconeogenesis, and so forth. And it is well appreciated that disruptions in the signaling pathway can have detrimental metabolic consequences, including insulin resistance, hyperglycemia, and in some instances, cancer. So what my lab is focused on are scaffold proteins, and specifically scaffolds belonging to the 1433 protein family. The classical definition of a scaffold, when you look in our biochemistry textbook, is a protein that anchors other proteins in a specific compartment of the cell. So here shown is the scaffold in blue, and it's holding these green proteins just under the cell membrane. Now, the role of scaffolds has evolved over the years, and now it's been shown that scaffolds can also protect other proteins from becoming dephosphorylated, as well as helping to form signaling complexes. And this is a concept that we'll get back to in a bit. Now, for those of you who are interested, that's a little shameless plug. My postdoc, Yves Mugabo, who may or may not be listening on this, he wrote a mini review paper in endocrinology, which describes the various roles of different scaffolds in metabolism. So here's just an example of a cartoon showing the contributions of the beta arrestins in metabolism. And then other scaffold families include KSR2, so the kinase suppressor of RAS, which has also been shown to have various metabolic outcomes or roles in the cell. So my lab is focused on 1433 proteins. These proteins were initially discovered in the brain, and they comprise about 20%, if not more, of all proteins within the brain. And they have their name due to the fact that they were discovered or identified from the 14th fraction that positions 3.3 of an elution of all brain proteins from homogenates. And what I find really fascinating about this protein family is their ability to bind to phosphorylated proteins harboring either serine or threonine motifs. And this really affords them the ability to interact with singly or doubly phosphorylated proteins. And this allows them to promote changes in the conformation of their binding partners. Sorry, someone just pulled up in my driveway. And also, they're able to regulate protein activity or stabilization. And one can actually envision that when you have these client proteins bound to 1433 dimers, they can also help to nucleate protein complexes that are critical to cellular processes. Now, over 1,800 proteins have been identified as 1433 protein binding partners. But what is important to note is that each isoform has specific binding partners, and that the interactome of all 1433 proteins is highly dynamic. So what I mean by this is that in the context of different physiological stimuli, or even pathophysiological stimuli, the interactome can change. And this can, in my opinion, open up new avenues of research to identify new factors that underlie physiological and pathophysiological processes. So my lab focuses on 1433 zeta. And what's interesting about this isoform and the other proteins is that they're actually known to have metabolic functions. But the caveat is that a lot of this work has been done in cell lines. So since starting my lab in 2016, I've extended on work that I did as a postdoc at the University of British Columbia. But before I get into that, just want to say that my lab really has two separate aims or research areas. One is to identify the role of 1433 proteins in adipocytes. So focusing, for example, on cell fate, thermogenesis, as well as just overall adipocyte function. And we also have a separate research aim or arm focused on the role of 1433 proteins in the pancreatic beta cell. And this is work that is spearheaded by a talented postdoc in my lab, Yves Mugabo. And congrats to Yves, he has an oral presentation at the ADA if it happens this June. And maybe in the future we can talk about this. Or if someone's interested and wants to find out more about the role of 1433 proteins in beta cells, just send me an email or DM me or even call me. So for today's talk, we'll just be focusing on 1433 proteins in adipocytes. So the reason why I really got interested in 1433 zeta specifically is that when I was a postdoc at the University of British Columbia in Jim Johnson's lab, we started to characterize 1433 zeta knockout mice. So these are whole body knockouts, and they're depicted here in the red triangles. And as you can see from weaning up to the endpoint of the study at 24 weeks of age, that the knockout mice weigh less than their wild type littermate controls. And this decrease in body weight was really because these mice had quite striking reductions in fat. So here's just a picture of two mice, wild type on the left and a knockout on the right at time of necropsy. And you can see that the fat pad here in the knockout mice is substantially decreased. And we can also quantify this using DEXA body composition analysis, and we found that this was associated with approximately 50% reduction in overall adiposity. Of note was that the fat pads that seemed to be affected were actually those related to the visceral depots. So for example, the gonadal and the perirenal depots. So knowing that the knockout mice were lean and had less fat, we were fortunate to get transgenic mice that overexpress 1433 zeta, and more specifically a tap tag variant of 1433 zeta. And we just put these mice on a high fat diet, and as you can see here in green, when the tap mice were fed the high fat diet compared to their wild type littermate controls, that the transgenic mice that overexpressed 1433 zeta gained significantly more body weight when fed the high fat diet. And this was due to the fact that these mice had an increase in fat mass. So to us, this was very exciting in that it showed that the loss and gain of function have completely opposite effects with regards to regulating adiposity. So a lot of this work is published. It's published 2015 now. But what we found was that 1433 zeta controlled or regulated the adipogenic program through the actions of GLE3, which is part of the hedgehog signaling protein pathway, as well as, and then in doing so, induce cell cycle arrest in progenitor cells. But as I mentioned earlier, that well over 1,800 proteins are known to be interacting partners with 14-3-3 proteins. So this really begs the question of whether other factors could also interact with 14-3-3 zeta to influence adipocyte development as well as function. So since starting my lab at the CR SHUM, we've really addressed this by using proteomics. So we hypothesized early on that, well, maybe we could find novel regulators of adipogenesis within the 14-3-3 zeta interactome. So to do this, we collaborated with Thibaut Mayor at UBC, and we took the same transgenic animals that overexpress 14-3-3 zeta, and we generated mouse embryonic fibroblasts from these mice, and just had two conditions: an undifferentiated group of cells, and then those that were stimulated to differentiate for 24 hours with the well-established cocktail of IBMX, dexamethasone, insulin, as well as rosiglitazone. And we took cell lysates and then did some pull downs and then did some mass spec to look at what interacts with 14-3-3 zeta in the undifferentiated and differentiated state. And over 120 proteins could be identified within the interactome 120 proteins were actually found to be enriched in cells that were undergoing differentiation. Okay, so we just basically did a subtraction of what was common to both conditions. And what was quite surprising was that within the interactome, we could find known regulators of adipogenesis. So for example, PTRF, which it's also known as CAVIN1, was found within the interactome of 14-3-3 zeta of cells that are undergoing differentiation, and that if we use siRNA, we could indeed knock it down, and also inhibit differentiation as assessed by LRED-O and PPAR gamma mRNA expression. Other known factors included the prohibitins, so prohibidin 1 and 2, which are known to be involved in mitochondrial function as well. So with 120 proteins, we were at a loss as to how to really interpret the data. So we just took a step back and just used gene ontology. And what we found was that the proteins that were highly enriched within the interactome were those related to RNA splicing. I'm sure I don't have to really explain this whole process to the audience, but RNA splicing is the event by which our pre-mRNAs, various exons are either omitted or skipped or joined. And this really is important in terms of increasing our proteomic diversity. Our genome is roughly 20,000, 25,000 genes. So clearly there has to be a way to encode for more proteins than our genes would indicate, right? So what Yves did up here is, this is his main project in the lab, or part of his main project is that he focused on PPAR gamma. So PPAR gamma, or PPARG, as many of you know, is the master regulator of adipogenesis. And it itself is spliced during differentiation such that the PPARG2 variant is the critical isoform that regulates adipogenesis. So what he used was quantitative real-time PCR. Oh, whoops, let me back up. So what he first did was we went through the Sigma catalog to look for spliceosome inhibitors, and we came up and found madrasin. And here you can see that if we use madrasin at a concentration of 10 micromolar, that he was able to block adipogenesis as assessed by LRED-O. And looking at PPARG2 splice variant generation, you can see that this spliceosome inhibitor does indeed significantly decrease levels of this variant, as well as this novel PPARG1 splice variant following differentiation. And these decreases in, so then what he did here was that he used siRNA against 14-3-3 zeta, and sure enough, he's also able to see that we're able to decrease PPAR gamma 2 protein levels, as well as the mRNA, which I'm not showing here, apologies. So we then said, okay, well, that's great. So what about the different splicing factors within the interactome? Can we identify novel factors or novel splicing factors that are critical for 3T3L1 adipogenesis? So he focused on these splicing factors here, and I'm go through them all, but we really just focus on these primarily because they were found to be the most displayed the highest degree of interaction with other factors. So essentially you could think of them as how well connected they are to the other proteins. And using siRNA, what you found was that not all of those splicing factors had effects on PPARG2 mRNA expression. Specifically, he found that HNR-MPF was one of the most important factors in terms of generating PPARG2 mRNA. And we could also see that this was associated with decreases in the protein when we measured it by Western blot. And sure enough, when we did oil red O, you can see that knockdown of these various splicing factors also inhibited adipogenesis at the endpoint of this study. So now Eve is carrying on this work in the lab and trying now to further explore the roles of HNR-MPF in adipocyte development. So what I've shown you so far is just one aspect of the lab, which is how does 1433 zeta regulate differentiation? And the other question that we're also interested in is, does 1433 zeta have any effects in mature adipocytes? And specifically with regards to lipid storage, lipolysis, and thermogenesis. Now, because I live in Montreal, for those of you who don't know, Montreal is in Canada. It's a pretty cold city in the winter. This is the morning of a snowstorm we had in February. And some days we wake up and it's minus 27 Celsius. And it feels like minus 33 with the wind chill. So it's, for some people who like being outdoors and in the snow, it's great. For some of us who don't, this can be torture, especially when this carries on for months on end in the winter. So just to show you an example, it's the aftermath of the snowstorm. This is my son. It's about 4 feet 2 inches tall, and the snowbank at the end the next morning was around 3.5 feet. So we get quite a bit of snow, right? So going back to this thing of living in a winter, right? Some of us do not like the cold, and obviously we have ways to defend against cold and to tolerate cold. So we have the ability to put on coats. Obviously, in the short term, we have mechanisms such as vasoconstriction as well as shivering, but both of these two tend to be short-lived. And if you're lucky, you can get on a plane and fly down to Mexico for a week or so just to thaw out. But obviously, this isn't applicable to everyone. But one way that, and this is something that has become of interest to the field for several years now, is the idea of non-shivering thermogenesis. So this is a PET CT scan showing, looking at 18-FDG uptake, and you can see here these specific depots of brown adipose tissue, as well as some organs like the heart, take up this radio-labeled tracer and indicating that you have activity of brown adipose tissue, which is known to generate heat. So what we know is that we can take brown or adipose tissue and really segregate them into two different types. So brown adipocytes, which are rich in mitochondria, and these are primarily involved in thermogenesis, or the generation of heat. And then we also have white adipocytes, which can be further divided into subcutaneous or visceral depots, in the subcutaneous or visceral locations. And the primary role of these adipocytes are to store lipids or to provide energy in the form of fatty acids in times of need. Now, it's become appreciated that activation of the sympathetic nervous system can also transform these white adipocytes into an intermediate form known as beige adipocytes. So these resemble brown adipocytes in that they become enriched in mitochondria, as well as they also exhibit a multilocular, so they have multiple smaller lipid droplets within the cytoplasm. And these adipocytes are thought to be involved in thermogenesis as well as lipid oxidation. So why what's so, you may be asking, well, what's so important with regards to having many mitochondria within these cells, and how does it lead to heat? Well, this is really stems from the fact that the mitochondria also have, and these adipocytes, have high levels of UCP1, so uncoupling protein 1. And this is a protein that uncouples the proton gradient during oxidative phosphorylation. So instead of making ATP, protons go through this channel or transporter and to generate heat. So the idea of the sympathetic nervous system and really the release of norepinephrine from the sympathetic nervous system has been known for quite some time. Here's just one example where of how norepinephrine may regulate thermogenesis is that you can take, this is a study done in 1957. They took rats and injected them with norepinephrine. And here you can see that there is quite an increase in oxygen consumption or heat generation. This is also reflected by an increase in the rectal temperature, right, of these rats. And we know that UCP1 is important for glucose utilization as well as thermogenesis, because this is a study using UCP1 knockout mice. If you take wild type mice and you inject them with norepinephrine and you measure temperature within the brown adipose tissue depot, you can indeed see an increase in temperature in bat from the wild type animals. However, this is lost in UCP1 knockout mice. And so there's, right, sorry. So the idea then that UCP1 is important in thermogenesis has been established, right? I think this is well established by many labs, as well as the role of norepinephrine. But what is actually interesting is that in addition to norepinephrine being able to stimulate thermogenesis, we also do have a balance or another system within our bodies that can have negative effects on thermogenesis. And this includes serotonin. So both norepinephrine and serotonin are synthesized by the activities of tyrosine hydroxylase and tryptophan hydroxylase. And these enzymes have been quite elegantly shown by several groups to be critical for thermogenesis. So this is an example. This is a recent paper from Christoph Buttner's group where they used, they generated tyrosine hydroxylase knockout mice. So these were whole body knockouts. And here you can see that when they, if they injected them, oh sorry, when you place them in cold, that you can see that these mice that lack tyrosine hydroxylase within every cell of the body, the peripheral tissues, that you can see here in the black that they are unable to mount an appropriate temperature response when placed in cold. With regards to serotonin, this is a really nice paper also from Greg Steinberg's group at McMaster, where they used TPH1 knockout mice. And here you can see that if mice that lack the ability to produce serotonin, that if they, these mice, sorry, where am I? Here's some mess. That these mice do indeed have a higher basal VO2, so oxygen consumption. And when these mice are injected with CL, which is a beta-3 adrenergic receptor agonist, that these mice do indeed have the ability to generate more heat. And this is done using a FLIR camera, so an infrared camera. So you may be wondering where I'm going with all this, right? And how this, whether this relates in any way to 14.3.3 zeta. So one of the initial roles of 14.3.3 proteins was actually their ability to regulate tyrosine and or tryptophan hydroxylases. So this is their alternative name up here. So we asked, well, does 14.3.3 zeta, because knowing that it can regulate both tyrosine and tryptophan hydroxylase, we asked a very simple question of whether this protein is critical for generating heat or thermogenesis in the context of cold. I want to stress that we're we don't know what cells are responsible, right? We just wanted to ask, does 1433 zeta, through its actions potentially on tyrosine or tryptophan hydroxylase, have any effects on cold? So this is work done by Khadija Diallo, who was a talented student in my lab and recently finished her master's. So what she did was that she first used an acute cold challenge model. So we took advantage of the wild type and heterozygous mice, 1433 zeta mice in our lab. So as you may recall at the beginning, I showed that knockout mice were lean, they weighed less. And so we did not want any of those confounding factors in our study. We wanted mice that had equal body weight, but only lacking one allele. So you can think of it as a gene deletion, right? And so what she did was she put the wild type and heterozygous mice and placed them in a cold room for 4 degrees Celsius for 3 hours. So before and after the cold challenge, there were no differences between genotypes in terms of body weight. And these are data from male mice, and as you can see that the heterozygous mice do not display any differences with regards to rectal temperature at the end of the study. And but what we did see, at least at the message level, with regards to UCP1, that at least in the brown adipose tissue, that UCP1 levels, mRNA levels were indeed going up when these mice were challenged with cold. So this was in some ways a bit disappointing because we thought, oh, maybe by losing one allele of YWHAZ, which is the gene encoding 1433 zeta, that we would indeed see some change in cold tolerance. So we then turn to the TAP mice. So once again, these are the mice that overexpress 1433 zeta. And it's relatively a mild overexpression, okay, at the protein level. But at the message level, so this is for human 1433 zeta, human YWHAZ. As you can see that these mice do indeed express higher levels at the mRNA level, at the human gene, and this is not present in the wild type mice. And Khadija did the same paradigm. So she put the wild type and TAP mice at 4 degrees for 3 hours. And once again, these are data from male mice. And as you can see here in the white curve here, that the TAP mice actually started to exhibit this improved tolerance to cold at the endpoint of this study. So this was quite surprising to us. And when she did some mRNA expression analyses, she found that UCP1, which encodes, UCP1 was indeed higher in the inguinal adipose tissue from the TAP mice. So with this observation that the transgenic animals seem to have this improvement in cold tolerance, we then decided to adopt a more chronic model. So what she did was Khadija took mice, wild type and TAP mice, and subjected them to this kind of experimental protocol. So she did echo MRI to look at body composition analyses. She also then implanted a temperature probe the day prior to placing the mice in the clams. So it's a comprehensive lab animal monitoring system. So it's essentially a metabolic cage. And here you can see, so she put these mice at 22 degrees, and then we just dropped the temperature in the unit to 4 degrees, and we left them there for 3 days. And as expected, that there was a decrease in fat mass in the wild type and TAP mice following chronic cold exposure. This makes sense because in times like this, the mice are trying to generate heat, so they are burning fatty acids through thermogenesis. And I guess the good thing about putting them in the metabolic cage is that we can generate a lot of data and different types of data. And so here you can see that the food intake at different times or different parts of the light or dark cycle at the different temperatures. For the most part, there, nothing was really different between groups. Same with locomotor activity or RER. But what was quite surprising was that when she then looked at body temperature, you can see here, so the TAP mice are shown in red, that the tap mice do indeed have a higher body temperature than the wild type mice. So we then looked at energy expenditure or heat generation. And this is where things in some ways took a turn, an interesting turn, and that the energy expenditure in the tap mice, for the most part, you can see with the traces averaged out, that is lower in the tap mice than in the wild type littermate controls, and that there was significance at these 2 stages of the dark cycle, right? So we were very puzzled as to why is it that body temperature is going up in the tap mice, but we're actually seeing an opposite decrease in energy expenditure or heat generation. Normally, one expects both of them to go up at the same to the same degree. So to explore this further, we just first did some more characterization of these mice following the cold challenge. We wanted to see whether there were any changes in adipocyte morphology, and long story or short answer is no, there were no differences between the wild type and tap mice with regards to the size or the distribution of adipocytes within the gonadal or inguinal depots. When we looked at the brown adipose tissue from the wild type or tap mice following the chronic cold exposure, we did see something interesting here. Happy to discuss this further and get some potential insight as to what may be happening, but you can see that the lipid droplets within the brown adipose tissue, so this is staining for perilipin, were actually quite small compared to the wild type littermate controls. Despite all this, there were no differences in triacylglyceride content among the depots that we tested, so the gonadal, inguinal, or brown adipose tissue depots. So Khadija then examined whether there were any changes in beiging of the inguinal adipose tissue. So, it is thought that by these brown adipocytes, these white adipocytes within the inguinal adipose tissue depots, it's thought that by them acquiring this beige phenotype that they could indeed or potentially contribute to thermogenesis. And this is something that I know that quite a few labs are studying. So what she first did was she looked at gene expression in the brown adipose tissue depots. And then she followed this up by looking in the inguinal adipose tissue from mice subjected to the 3-day cold challenge. And here you can see that indeed UCP1 mRNA levels are indeed going up in the tap mice, which is suggesting that beiging is indeed happening in these depots, in the inguinal depot. And she then did some histology. So we did some immuno staining for UCP1. This is their inguinal, from inguinal adipose tissue sections. And you can see that indeed there's quite a robust increase in UCP1 immunoreactivity in the tap mice. And then by Western blot, you can see that at the protein level, that UCP1 levels are indeed going up. And there's actually, and also an increase in terms of UCP1 protein content in the brown adipose tissue from the tap mice as well. Of note, we do see an increase in FGF21 mRNA in the inguinal adipose tissue depot in mice following the chronic or 3-day cold challenge. But this is something that we didn't really go explore further. So I then asked Khadija just to check whether there were any differences in between groups to adrenergic or beta adrenergic stimuli. So we adopted this paradigm here where we took wild type and tap mice and injected them daily with the beta-3 adrenergic agonist, the CL compound. And before and after CL administration, we did some echo MRIs to look at body composition. The long story short is that there were no differences between groups. So no changes in body weight, and also no differences were observed with regards to the induction of UCP1 mRNA levels between groups following the CL exposure. And then looking at tyrosine hydroxylase itself within the different depots. So she did Western blots for TH and the IWAT and BAT. Really nothing were different, nor were there differences in the tissue content or norepinephrine. So, you know, we know that full and cold exposure, there are changes in glucose utilization. And, you know, one of the, I guess what's relatively easy for us to do was to just take mice and put them in a cold room again for 3 days, and then look at whether there were changes in glucose utilization or really glucose tolerance between groups. Here you can see that, you know, compared to the wild-type litter meat controls, that the tap mice appear to have an improvement in glucose tolerance. However, this change in tolerance was diminished full and cold exposure. Nonetheless, we decided to explore this a little bit further. So we started a collaboration with Andre Carpentier at Sherbrooke, and we took advantage of his unique PET-CT system or facility there. And so what we did was we sent him wild-type and tap mice, and we subjected them to a randomized crossover study. So groups of mice were either housed at 4 degrees or thermal neutrality for 3 days. We did a round of PET-CT imaging, specifically looking at C11 acetate and 18-FDG. So C11 acetate will measure oxidative, it's a measure, can help to measure oxidative metabolism, and 18-FDG is just really glucose uptake. And after the initial PET-CT run, we let these mice recover for 7 days at room temperature, and then subjected them to the different opposite temperature for 3 days, and followed this up with PET-CT again. So these data are preliminary, and the last cohort of mice are being, the data from the last cohort of mice are being analyzed. So what we found was that between groups, or even really between temperatures, there were no differences in oxidative metabolism in the heart, liver, or kidneys of these mice, nor were there really any differences with regards to the genotype or effects of the genotype on 18-FDG uptake in the heart or kidney. We then looked at the inguinal or brown adipose, or inguinal visceral, which is the gonadal depot in this case, and also brown adipose tissue. We then looked at 18-FDG uptake, and really for the most part, no differences. When we looked at oxidative metabolism, same thing, no differences between brown adipose tissue, in the brown adipose tissue between groups, or in terms of temperature. We tried looking at the inguinal white adipose tissue from these mice, but it was actually undetectable. And this is something that I think it's, you know, it's been shown now by us, or we're reporting this now, and then also shown by others, that it is actually very hard to look at oxidative metabolism in IWAT, presumably due to the fact that there's just very few cells that are have undergone beiging, if you will. So let's take a step back and, you know, sort of address what I've shown so far, that there is no change in, or chronic cold exposure leads to an increase in body temperature of a tap mice, but we're not seeing, if anything, we're seeing an opposite decrease in energy expenditure. There are no differences in sensitivity to beta adrenergic stimuli, and potentially there are no differences in brown and or beige adipose tissue function. So what could be accounting for this, these changes in body temperature and energy expenditure, right? So we took advantage actually of this paper that was published in Molecular Metabolism from the Morton group. And herein they report, utilize this concept of thermal conductance, right? Which is actually a measure of heat loss or heat dissipation. And we were able to calculate this because we already had our metabolic cage data. And so what Khadija then did was, sure enough, what we see is that if you look at thermal conductance, so heat loss, the top mice shown in white, whether these mice are housed at 22 degrees or at 4 degrees, they actually seem to have a decrease in the amount of heat loss at any temperature tested. And although this paper looks at the effect of leptin, we don't see any changes in these mice in terms of circulating leptin, but we did not challenge them in any way, okay? Now, what could be accounting for this change or decrease in heat loss or heat dissipation? We took advantage actually of, well, we were inspired by this paper from the Cannon Niedergard labs where they looked at the tail of mice and measured temperature drops in response to leptin. But the reason why the tail is actually quite a useful site to look at temperature is that, at least in rats, obviously, and in mice, the tail lacks fur. And with respect to rats, it comprises 7% of the body surface area. And under steady state conditions, the tail actually loses about 20% of body heat, and it is highly vascularized. So we then collaborated with Alain Rivard down the hall from us, and he has a laser Doppler setup to look at blood flow. And sure enough, what we do see in the tap mice is that there is overexpression of 14.3.3 zeta seems to decrease blood flow, which one can then potentially correlate to a decrease in heat loss. Right, so to sum this up, you know, what we know in terms of a wild type mouse is that if you put them in cold, obviously you have an increase in heat production and energy expenditure, but there's also quite a bit of heat loss from the tail and even to some degree from the paws. And what we're showing is that if you have mice that overexpress 14.3.3 zeta, yes, you do see an increase in heat production, but this is associated with a decrease in energy expenditure and heat loss, presumably due to the fact that we have an increase in vasoconstriction in the tail. And I think what these data are quite helpful in that they, you know, they tell us that in terms of cold tolerance, one should not always focus primarily on thermogenesis, let's say from brown adipose tissue. I think it's important to consider all of the other factors or physiological mechanisms that can regulate temperature. So in the last bit of this presentation, I'm just going to share with everyone another study from the lab quickly. So before in Khadijah's study, we looked at whether 14.3.3 zeta affects thermogenesis or cold tolerance. You know, and in that study, we're not really looking at the adipocytes themselves. So, you know, we're taking advantage of the genetic tools available now to ask the question of whether 14.3.3 zeta can directly influence adipocyte function. So this is the work of Abel Opong, who was also a master's student in the lab and graduated around the same time as Khadijah. And what he did was he generated adipocyte-specific 14.3.3 zeta knockout mice. So here we use the Adipocrete ERT2 from generated by Stefan Offermans and bred them to mice with flox alleles of YWJZ. So to generate adipocyte-specific 14.3.3 zeta knockout mice. So just to show that indeed we are able to decrease mRNA levels of YWHAZ. This is from whole adipose tissue. So one would assume that the residual message would be from the other cell types within the adipose tissue depots. There were really no effects on body weight when we knocked out 14.3.3 zeta, nor were there any differences in glucose tolerance or insulin sensitivity in these mice. But what Abel did observe was that when he injected mice with CL, so the beta-3 adrenergic receptor agonist, you can see that at least in terms of glycerol, that these mice do have an impairment in lipolysis, and that circulating levels of glycerol in the knockout mice were lower than in the wild-type littermate controls. He further explored this using gonadal white adipose tissue explant studies. And here you can see that both glycerol and free fatty acid levels were significantly decreased in the knockout tissue. So we did some more phenotyping of these mice, and at least at the gonadal white adipose tissue depot, you can see that there was a change in the size distribution of the adipocytes within this depot, such that we seem to have an increase in, a decrease in adipocytes that's smaller than roughly 200 micrometers, and then an increase in larger adipocytes. Now, I then asked Abel to really look at whether, are there other changes in terms of adipocytes within following 14:3:3 zeta knockout that could help to explain potentially why is it that these mice have an impairment in lipolysis. So what he did, well, among different experiments that he did some gene expression studies looking at HSL as well as PPARG at both the message and the protein level. And quite surprisingly, so this is from the gonadal white adipose tissue depot, that at the protein level, it appears that both HSL and PPAR gamma 2 protein abundance were significantly decreased when 14:3:3 zeta was knocked out. So to us, it suggested that maybe there was something going on in terms of the maturity or the status of these cells following 14:3:3 zeta depletion. And when he then did some qPCR to look at the so-called mature adipocyte genes, sure enough, deletion of 14:3:3 zeta does indeed decrease, genes such as adiponect or adipoQ and resistin in both the GWAT and the IWAT, as well as with regards to triglyceride synthesis in the gonadal depots. Some of these genes which are required for de novo triglyceride synthesis are decreased. Now, I then asked Abel to look at sort of the expression or localization of 14:3:3 zeta in these adipocytes. So looking at the gonadal depot, what was quite surprising was that when he did some immunostaining for paralipin to look at the lipid droplets to identify adipocytes and 14:3:3 zeta, you can see here the red arrows that there seems to be this heterogeneous expression of 14:3:3 zeta within the gonadal white adipose tissue depot in the wild-type mice, okay? And in our knockout mice, we do not see this heterogeneous expression, which is good, because it's showing that we are able to recombine and delete 14:3:3 zeta. So this actually, in some ways, made it very difficult to really characterize what could be happening, because it's tough now to really purify populations of cells that do or do not have 14:3:3 zeta. So Abel then went back to the 3T3L1 cell line, where he was able to induce quite a robust depletion of YWHAZ, which is the gene encoding 14:3:3 zeta. And then we can also see this decrease at the protein level when he examined this by Western blot. So what he also found was that when he knocked down 14:3:3 zeta, that he was also able to impair lipolysis in response to various lipolytic stimuli, which is in line with what he found in his adipocyte-specific knockout mice. And that although he did see changes in ADRB3 mRNA, which is which encodes the beta-3 adrenergic receptor, no differences in cyclic AMP generation were found when 14:3:3 zeta was depleted. Just like with the adipocyte-specific knockout mice, that HSL at the protein level, at the very least, was decreased when 14:3:3 zeta was depleted, as well as another component of the B3 signaling pathway, CREB. So Abel then went to look at what happens to 14:3:3 zeta, or what does 14:3:3 zeta do with respect to lipid content. So first, using oil red O, you can see that there is a small but significant difference or decrease in absorbance of oil red O. And here's just a microscopy showing, looking at the droplets themselves. Then this was done by Bodippy imaging. And here you can see that depletion of 14:3:3 zeta leads to quite a substantial decrease in the size of droplets in these cells. And when triacylglycerides were just measured biochemically from 3T3L1 cells that were depleted of 14:3:3 zeta, shown in black, that triglyceride content was significantly decreased as well. So these different or decreases in lipids were also associated with decreased mRNA levels of various genes associated with de novo triacylglyceride synthesis, much like what we found with our adipocyte-specific knockout mice. And lastly, we also found that genes associated with adipocyte maturity were also significantly decreased in the 1433 zeta depleted 3T3L1 cells. So to us, this suggests that 1433 zeta in some ways is required for maintaining a mature adipocyte phenotype. How it does this is at this point still up in the air, and we have some ideas, but obviously due to current conditions, we're unable to test them or explore them further. But what I will say is that we took advantage also prior to the shutdown of collaborating with Christine DeRosier at the Montreal Heart Institute to do some lipidomics. So we wanted to see whether, could we in some ways generate a lipidomic signature that would be associated with immature adipocytes. So what we did was we took 3T3L1 cells, electroporated them with siRNA against 1433 zeta, waited 48 hours, and then did some lipid extraction and then LCMS to identify the species. And it's actually quite interesting. So what we found was that over 500 lipid species were altered. Shown here are the various species that were identified with the in-house annotation database. And just by looking at the types of triglycerides, for example, you can see here that many triglycerides that were a shorter acyl chain length or carbon chain length were significantly decreased, as well as those that had lower degrees of saturation. Now, we took advantage of a paper published a few years back looking that reported what happens in terms of triglyceride species in the context of differentiation, and specifically in 3T3L1 cells. And what we found was that species that should normally be higher or enriched and differentiated cells, so for example, triglyceride species of 48 carbons and 3 degrees or 3 saturations, that these species were actually decreased following 1433 zeta knockdown, whereas species that would normally be decreased in undifferentiated cells were actually increased. So, we can take from this that perhaps 1433 zeta is required for maintaining not only the gene expression profiles, but also this so-called lipidomic signature as well. So just to summarize the talk, I hope, at least I've conveyed today, that 1433 zeta has multiple roles within adipocytes. It stems from regulating adipocyte maturity as well as differentiation. And it may also have a role in terms of thermogenesis and heat regulation. Obviously, these do not indicate that they are cell autonomous roles within the adipocyte, but at least 1433 zeta may be acting on other cells or influencing other physiological mechanisms in the context of thermogenesis. So the work which I presented to you today are both in preprint form, so Khadijah's work, some bioarchive, as well as Abel's. Obviously, happy to answer questions after the talk via Twitter, but please feel free to send me an email or a message or call me if you want. We can discuss this further. Obviously, this is a picture of, I'd like to thank the people in the lab who did do the work, as well as the current members from here. This is the picture from last summer. And last but not least, I'd like to thank the funding agencies for supporting this work. Yeah, so happy to take any questions. And thanks for the opportunity and thanks for listening.